Hello, today we're going to be teaching you everything you need to know about DNA synthesis. So DNA synthesis is where our DNA technically, replicates. There's a few big things in here that is kind of a major thing we need to know before getting into this. So the first thing is the enzymes, anything really that ends with ASE. We'll talk about what they are and all of their specific jobs, but they can be related to, really anything else. We'll get to that as we go on. But you also need to know that new DNA strands can only be connected to an open 3' end. So these numbers will mean something in a second. So you can see our DNA strand we have here. Our top side is a 5' and our bottom side is a 3'. And so these are just interwoven. Think about it like a zipper. A zipper, when you zip it up, it fits each other perfectly like a little puzzle. And that's what we have here. And so the first step in DNA replication is this right here. This is our actual zipper. This is the enzyme helicase. And the area where it unwinds the zipper, right here, that's called the replication fork. The replication fork. And so both of these will actually move that way as DNA replication occurs. So now you can see that we have two different strands here. This top one is going to be our leading strand. The bottom will be our lagging strand. The names will mean more in just a second. And so the top will end in a 3' and the bottom will end in a 5'. Get it? Because it is the opposite of what the strand started as. And so now that we know DNA can only be created at an open 3' end, that's where we see our leading strand. It's got a much simpler task because now that it's a 3' end, the enzyme DNA polymerase acts as a sort of a builder. So we have our zipper here. So when a zipper gets unzipped, well, now you have those teeth that are kind of sticking out, and they need, in order to really become whole again and to actually do their job, they're going to need a pair to go with it. So we have our open zipper teeth here. And now you think about DNA polymerase as kind of being like a creator of a new zipper that is going to come on the back side. So DNA polymerase will create a strand that will continue and actually match the zipper teeth we have up on the top. So now we have a zipper that, as the helicase unzips, the DNA polymerase will just keep re-zipping and creating a new strand. The bottom is a little bit different though. Since it ends in a 5', we cannot do the exact same process we did on the top. We have to work backwards. So it actually has to start with the enzyme, the DNA primase will actually create the first few strands of our bottom teeth. So this is the DNA primase. And so that acts as sort of a primer where then DNA polymerase can come in and connect itself right here, and then it does its job. It can create the next few in that order. Oopsies. It will create the next few in this order right here. So it does the next few teeth. But it's not able to do all of them. There's still a gap right here where these last two teeth need something to happen. And so that's where an Okazaki fragment comes in. So the Okazaki fragment will come and collect itself right here. Okazaki fragment. But you can see there's still a gap right here between our Okazaki fragment and where our DNA polymerase left off. That's where DNA ligase comes in and joins the two together. And so now we have just matched this strand. But the issue is, as this keeps unwinding, this exact process is just going to keep happening over again. That's why it's called the lagging strand. As it unzips, it'll build something, but then it's got to go back and build more. Whereas the top continues to build, but because of the open 3' end, it's going to just have to keep the open 5' end, it's going to have to keep making this exact process happen over again. And so all of this right here is going to be technically the Okazaki fragment for the next part of the DNA replication chain. And so that is basically quick everything you need to know about DNA synthesis.